Hi guys, so let's continue with our biochemistry lectures. We are discussing glycolysis. Glycolysis literally means breakdown of glucose, cutting glucose into two pieces. Now we know that glucose is 6 carbon sugar. We are breaking into 3 carbon molecule. Now this glucose is coming inside the cell, okay, by glut. This is glucose transporter, and this glucose transporter is carrying glucose inside the cell. Now there are high chances that the glucose can go outside of the cell also if the concentration is more outside. Okay. So to make glucose stay in the cell, we need to do something. Okay, we need to do something to the glucose so that it doesn't run out of the cell, because cell needs the glucose. Right? So what do we do? And I am representing glucose by this structure. This is 6 carbon, so this is first carbon, second carbon, third carbon, fourth carbon, fifth carbon, sixth carbon. Okay, of course we can draw glucose like this also, the real molecular structure of glucose, and it's also very easy, something like this. Okay, we have O group. Okay, then we have okay, and then we have here, this is our glucose structure. Okay, this is our glucose structure. So this is first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. This is first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. So this is the glucose molecule. Okay, now if we need to change this glucose, so this glucose do not go inside. So what do we do? So we have very interesting enzyme. Okay, so what enzyme it is? Let us see. So if we put, let's say this is 1, 2, 3, 4, 5, 6 carbon, and if we put one phosphate group which is charged group, let's say I am putting here a charged group. Now we know this charged molecule cannot pass the membrane very easily. Okay, so what we have done? We have added one phosphate group to the glucose. So if we see in this structure, what do we do here? This glucose is converted into, we will draw glucose because glucose structure is not been changed. It is similar. We are just adding one phosphate group. Everyone should at least know how to draw glucose, and it is very simple to draw glucose. Okay, so this is the glucose, and what we are doing, we are adding phosphate group here. Okay, we are adding a proper phosphate group over here. So how we add a phosphate group over here? We will add phosphate group at this position. Okay, we will add phosphate group at this position, which is 6th carbon. See, here also we have added phosphate at 6th carbon. Okay, so the enzymes which transfer the phosphate group, what are those enzymes called? Those enzymes are called kinases. Okay, so we have very interesting two enzymes over here, not two enzymes specifically. So let us talk about one enzyme first. So we have one enzyme which is, let's say we name that is a Honda enzyme. Do you know bike, Honda bike? So like that we have one enzyme over here. So this is, I don't know how to make a bike. So yeah, so this is Honda bike. Okay, this is the one enzyme. And what this enzyme is doing? This enzyme is making glucose into glucose 6-phosphate. Okay. Now this step is very important, guys. Why? Because this step is irreversible step. This step is irreversible step. This Honda enzyme, I'm just calling him Honda for a time being. This our bike enzyme, which is Honda bike enzyme, it cannot convert glucose 6-phosphate to glucose back again. This reaction will go only in this direction, only in glucose to glucose 6-phosphate direction. Because why? Because delta G over here is high, is very negative, extremely negative. So this reaction will move only in this forward direction. Okay, this enzyme cannot put this enzyme, flip this or make glucose 6-phosphate in to glucose. Of course, there are some another enzymes which can make glucose 6-phosphate to glucose, but this enzyme cannot do that. Okay, so this enzyme is very important. Okay, now there is one more enzyme over here we should of course talk about. Now this is Honda enzyme. Specifically in liver we see this kind of enzyme, and this another enzyme is, let's say these enzymes are like a huge jumbo bus enzyme. You know this jumbo bus, or you can also call, yeah, so jumbo bus enzyme. So this is big bus. This is big bus. Okay, this is big bus enzyme. So one we have Honda enzyme and one we have big bus enzyme. So why are we talking about this? Now you have to understand. See, if we talk about Honda bike, suppose you have, let's say you have a 2-liter petrol. Okay, you just have 2-liter petrol. Do you think bus will run very nicely or smoothly, or bus will run at very high capacity? Is it possible? No. Is it possible if you have 2-liter petrol, you can run the bike very well at a peak speed? Yes, you can. Okay. Similarly, this bus, okay, if you talk about this bus, okay, this is, let's not talk about the bus, let's say this is the big Airbus plane. You know, there are big planes, or jumbo bus, jumbo planes over here. So this is jumbo plane. Okay. Now this jumbo plane, do you think this jumbo plane will run in 2 liters of petrol? No, correct. You need lots and lots of quantity of petrol to at least make this enzyme start, to make this plane start. And once it is started, what is the speed of that? What is the speed of this plane? It is much higher than our bike. And also the carrying capacity of passengers is greater in your Honda or greater in your plane? Of course, greater in the plane. Correct? So let's say if we have just 2-liter petrol and if just have 2-3 passengers, or legally speaking only 2 or 1 passenger with 1 driver, of course the best vehicle is to get this bike running. But if we have just 2-liter petrol and 2 people to travel, do you think you will able to go and board the plane? No. Exactly like that, we have 2 different enzymes working for the same reaction. The same reaction is, the reaction here is to convert glucose to glucose. This molecule is called, what is this molecule called? This molecule is glucose 6-phosphate. This molecule is called glucose 6-phosphate. This glucose 6-phosphate, okay, has acquired a phosphate now, and it is charged, it is active. Now this glucose, it cannot convert back into glucose. And this has happened because of the 2 different enzymes which you are talking about. One is this bike and one is this plane enzyme. Let me tell you the name of this. Okay, so what I was talking about, just before telling the name, I guess you know the name, correct? So just before talking about this, the names of the enzymes, you should also learn how they act. Okay. So as I told you that where the demand is more, okay, for example, if you talk about liver, okay, let's say we have a liver over here. We have a liver. You know from GIT, as we have discussed in the last lecture also, GIT, all the glucose is taken in the liver and some amount of glucose is sent back into the circulation. And when there is blood glucose level is decreasing, liver sends some amount of glucose back into the blood circulation. Okay, so who is receiving high amount of glucose in bulk in short period of time? Liver. Correct. Which enzyme should work like that? Which vehicle you do think you should work? The bus or the Honda? Do you think if there is a Honda car here and there are only this much, like some millions and millions, or sorry, hundreds and hundreds of people are coming, do you think you can convert them with this small bike? No. You need a big airplane, you need a big Airbus. Exactly like that, we need this enzyme over here. Okay, and if there are simple cells, from blood, there is limited amount of glucose which is coming always. So which enzyme should work here? Do you think your plane should work or bike should work? Of course the bike should work. So the name of this bike is, the Honda bike is hexokinase. Hexokinase. Okay, and one of the isomers of hexokinase, isomer of hexokinase is your, I'm sorry, we should not call it isomer, we should call it isoenzymes of hexokinase is glucokinase. Glucokinase. So we have hexokinase and we have glucokinase. Now, when we talk about glucose to glucose 6-phosphate, specifically when we have high carbohydrate rich meal, when we have high glucose rich meal, of course the high amount of glucose is coming from the GIT to the liver, and then your glucokinase should work at that time. Correct? Glucokinase should work at that time. So we have to understand that how hexokinase and glucokinase works. Correct? Now, if you talk about, now you have understood which cells will use this enzyme. So majorly glucokinase is found in your liver, in your hepatocyte. Okay, and of course I am not saying that liver do not have hexokinase, it has hexokinase also, it has glucokinase also, but glucokinase is acted when there is high amount of glucose. If you draw a graph, okay, if you draw a graph of this both, okay, let's say this is the graph, okay, and this is the graph, and in this graph, let's say here you have, okay, here you have performance. Performance means you can also call it the rate of making it, the rate of making product, or the velocity. Okay, so this is a performance of this enzyme, and this is concentration of glucose. Okay, now here glucose is the substrate for this enzyme, so this is substrate concentration. So when we talk about hexokinase, our bike guy, okay, our bike enzyme, what will happen to this bike enzyme? Now, as glucose is increasing, the activity is increased. But suppose there are 10 people, do you think this bike will able to work now? No. The bike activity is full. Okay, bike cannot travel with more people, more than 2, or maybe in worst condition just 3. Okay, so when glucose concentration increase even little bit the glucose concentration is increasing, the performance goes flat. Okay, the performance goes flat. So this is the maximum performance by this enzyme. Maximum 2 people. Okay, maximum only this much amount of glucose it can convert into glucose 6-phosphate. Okay, so the performance is here. Okay, so this is what we call as Vmax. So this is maximum velocity what this glucokinase, this is our bike guy. Let me draw this bike. Bike guy can achieve, which is hexokinase. So hexokinase, even small amount of glucose is increased, it reaches its highest velocity which it can achieve, highest performance which it can achieve, and then it will go flat. Okay, so this is your Vmax attained by hexokinase. This is hexokinase. Okay, hexokinase. If we talk about glucokinase, let us represent it by a red color. Now, you know that glucokinase will not start in small amount of glucose. So he will struggle. He will struggle. But once he starts, he will convert huge amount of glucose into glucose 6-phosphate, and to reach its maximum velocity, to reach its maximum performance, you require huge quantity of glucose. Correct? So this Vmax will be achieved at very later stage than what we have seen for hexokinase. So this is for our Airbus. This is for our Airbus glucokinase GK. Okay. Now there is one more parameter we should see. Okay. There is something called Km. Km is the value. Okay. Km is the value. Km is the concentration of substrate at which half of the enzyme, okay, which half of the enzyme is saturated. So if it is getting fully saturated, let's talk about hexokinase. If hexokinase is getting fully saturated here, सब hexokinase खत्म हो गया, capacity खत्म हो गया hexokinase का, correct? At this concentration of glucose, hexokinase, this concentration of glucose, hexokinase is all saturated. So इसका half will be your Km. This is your Km. So if we plot here, so this is Km. And if you talk about Km of this glucokinase, see here it is not still achieved Vmax, or maybe it has achieved Vmax over here. So iska half. So iska Km, let's say यहाँ पे कुछ आएगा. So this is Km of glucokinase. Okay. So what we are seeing over here? If we talk about hexokinase, if we talk about hexokinase, let me write short form over here. If we talk about hexokinase have, yes, it has low Km. It has low Km, and what about Vmax? It has low Vmax. Similarly, if you talk about glucokinase have high Km and high Vmax. Correct? So if we are talking about saturation point, okay, let's talk about hexokinase. Okay, if we talk about hexokinase is getting saturated when glucose concentration is very slightly increased. Correct? थोड़ा भी ग्लूकोज आया, हेक्सोकाइनेस पूरा काम कर जाता है. Correct? So the affinity towards glucose, if you talk about the affinity towards the glucose, is more of hexokinase. Because see, glucose थोड़ा भी बढ़ गया, hexokinase का आधा concentration is done. Okay, half of the concentration is saturated. Correct? So the affinity, if you talk about hexokinase, so the affinity of hexokinase towards glucose is increased. The affinity towards glucose of glucokinase is decreased. Are we understanding what is we are talking about? Glucokinase and hexokinase. So we have talked about the reaction, we have talked about the enzyme mechanics, okay, about Km and Vmax of hexokinase glucose. And this one slightly more we have to add over here. Okay, what is this we have to add? Let's talk about the regulation, because with glycolysis we have to learn the regulation of glycolysis. Okay, so when we talk about regulation of glycolysis, just a minute. When we talk about regulation of glycolysis, okay, now regulation of glycolysis means regulation of this enzyme. As this steps is irreversible, you have to understand there are some steps in glycolysis. Okay, in those steps, the most important steps which are highly regulated, okay, let me repeat, in the whole glycolysis pathway, the most important steps which are highly regulated are those which are irreversible steps. In that, one is hexokinase and glucokinase. In that, one is glucose to glucose 6-phosphate. That's why I'm taking so much of time to discuss this step, because it's irreversible and it is highly regulated. So let's understand what it is, the regulation of glucokinase and hexokinase. See, if you talk about regulation of glucokinase and hexokinase, we have to understand who will regulate these enzymes. Okay, let me draw here again. So this is our bike. I'm just drawing bike like this. Okay, this is bike, which is hexokinase. इसको थोड़ा सा अच्छा बनाते हैं bike enzyme. So एक इसका सीट भी है. This is bike enzyme, and this is our jet enzyme. This is our jet enzyme, which is glucokinase. Now, who regulate this enzyme? If you talk about, see, every most of this irreversible enzyme has two kinds of regulation. One is the intrinsic regulation, okay, which is the for example, the allosteric changes which are brought by the nearby molecules, okay, and when there is extrinsic regulation which is done by some hormones. Okay, so let me show you. So this is our hexokinase and glucokinase. So let's talk about hexokinase and glucokinase. Both are kind of similarly regulated. Okay, so if you talk about regulation over here, okay, who is the activator of this? When do you need this to happen? So activator for this is, I will draw activator by let's say red, blue color. He key marker in blue color, so we will draw the blue. So we have one pocket. So activator, there is one guy over here which is AMP. AMP. If in the cell AMP concentration is more, so of course here that guy will be also activating. If AMP concentration is more, then okay, if AMP concentration is more in the cell, these enzymes will be activated. You know, in the start of metabolism lecture, we have discussed that AMP to ATP, ATP to AMP ratio. So, in the cell, if AMP concentration is very less, AMP concentration is very high, okay, because there are lots of AMP. Lots of AMP means there is less amount of ATP, correct? So, if there is less amount of ATP, that means cell is kind of energy deficient. So we need this glycolysis to run somehow. So this AMP is one of the activators of these enzymes. Okay, who is the activator of this enzyme? AMP is one of the activators. And this enzyme can be inhibited by this molecule. Which is this molecule? Glucose 6-phosphate. This can inhibit this molecule. This can inhibit these molecules. The glucose 6-phosphate can inhibit this hexokinase and glucokinase. It can inhibit it. Okay, so it makes sense, correct? If there is lots of glucose 6 phosphate, why to make more glucose 6 phosphate? Let us inhibit that. So inhibitors are glucose 6 phosphate, and activator is your AMP, correct? Now there is one more activator. Let's say this is the cell, and in the cell, let's say in the blood, your blood glucose level is very less. Okay, I will explain you more on this when we talk about some other reactions. Okay, I'm just introducing it to you right now. Okay, suppose the blood glucose concentration is very less. If the blood glucose concentration is very less, there is a hormone which comes and gets activated. Okay, this hormone is present in your pancreas. Which is the hormone in pancreas? If you are saying insulin, of course insulin is there in pancreas, but insulin is not secreted when there is low blood glucose, correct? When there is high blood glucose, that time insulin is secreted. But when there is low blood glucose, there is one hormone which is called glucagon, which is secreted. Now let's say this is our hormone called glucagon. Glucagon has a receptor, a 7-pass receptor. Okay, glucagon has 7-pass receptor. This glucagon will bind to this receptor. Okay, there is a G protein coupled receptor. This is a G protein coupled receptor. This is attached by GS type of protein. This G stimulatory protein will go and activate adenyl cyclase. Adenyl cyclase will convert ATP to CAMP. CAMP will activate pKa. Okay, CAMP will activate pKa, and this pKa, okay, and this pKa will come here, and will make glucokinase and hexokinase inactive. See, blood glucose level is less. Do we need our cells, specifically the cells, okay, which can obtain glucose from some another sources? Like you can make glucose from fatty acids, you can make glucose from amino acid. We will discuss that in coming lectures. But if there are mechanisms, okay, suppose and this cells, let's say we are talking about liver cell. Liver cell can make glucose from different non-carbohydrate sources. Liver cell can make glucose from different sources. Do you want liver to do glycolysis do you want glucose to make this glucose into glucose 6 phosphate and push it into glycolysis? We don't want that, because bahar, in the outside, in the blood, we need glucose over there. See, liver is like a caring mother. Okay, liver thinks about blood glucose first. Okay, if blood glucose level is high, he will take this glucose and it will convert into ATP and different all molecule. It will store in form of glycogen and everything. And when it is necessary, it will give this glucose to other parts of the body, into the general circulation. Liver cares about everyone. Exactly like that. If there is low blood glucose and there is one signal which is coming, which is glucagon saying, hey liver, blood glucose level is less. Don't spend glucose in glycolysis now. If you want energy, take energy from different sources. Give this glucose to blood. Give this glucose into the blood, because there are vital organs which are needing the glucose right now. So exactly like that, when blood glucose level is less, your glucagon comes, and glucagon inactivate hexokinase and glucokinase, and glycolysis will kind of stop there, or it will slow down at least. We'll see one more step which is we'll see one or two more steps which are very crucial in this. And there also glucagon acts, no doubt about that. So you understood that how it is regulated. So glucagon is the extrinsic regulator of this enzyme. Okay, glucagon is extrinsic regulator of this enzyme. Are we understanding? There is one more thing about glucokinase which I forgot. Okay, see, if you talk about bikes, where do you keep your bikes? You keep your bikes near your colonies or near your houses. Okay, but do you keep your bus like that? No. If you have bus, you will keep that in special kind of spaces. You need special kind of spaces. You need special type of caretakers to take care of this plane. Okay, this big Airbus. So like that, these glucokinase are present in the liver. Are present in the nucleus of the cell. Let's say this is the nucleus. I am just drawing here nucleus. Okay, in this nucleus there are specific proteins. Okay, let's say this is the gene. This gene will give rise to mRNA. This mRNA will give rise to protein, and this protein is very important. Okay, this protein is glucokinase regulatory protein. Glucokinase regulatory protein. This glucokinase regulatory protein regulates glucokinase. Okay, this glucokinase regulatory protein regulates glucokinase, and if this regulatory protein are activated, only then glucokinase will be activated. Okay, so glucose kind of activates it. Okay, so this so there are the same allosteric inhibitors and activators which will also regulate glucokinase activator protein. Okay, and this glucokinase activator protein will go and activate your glucokinase, and this glucokinase will do its job of doing glucose to glucose 6-phosphate. So you understood? So if we kind of write somewhere, okay, let me write over here. So which are the if we talk about regulation of this, okay, if you talk about regulation of this, so which are activators? Activators AMP, inhibitor is glucose 6-phosphate, and which is extrinsic regulator. Extrinsic regulator is glucagon. Glucagon inhibits hexokinase and also amount of glucokinase. Okay, so we are clear about the first step. First step is very simple, glucose to glucose kinase. We have talked about glucagon. See, glucagon receptors are not at every cells. When we talk about extrinsic regulator, glucagon receptors are not at each and every cells. These are present on very major. So major amount of glucagon receptors are present on your liver. Okay, in your liver. So liver has lots and lots of glucagon receptor. So liver will be more responsive towards glucagon. Not the other cells will be responsible for glucagon. Okay, so this extrinsic regulator will be mainly working in your liver cells and also some of the other cells. Okay. Yes, so this was about the first step. We will learn about more steps in the future videos. Thank you very much.